This podcast is sponsored by Scispot, the best tech stack for modern biotech.
One of the biggest challenges in global health is the fact that we have been blessed with antibiotics, and antibiotics have been a miracle cure. They've saved untold millions of lives over the years, right? However, it's an arms race with bacteria. Bacteria, evolve resistance against antibiotics, and so now we're facing a situation where a lot of the antibiotics that we used to use all the time, most bacterial strains out in hospitals and out in the world are resistant to them.
Right.
And, think about imagine a future where, every operation could be your last one, where you can get a scratch on the, like outside somewhere, you can get a scratch or you're doing sports or something, and that gets infected and you're dead because antibiotics don't work anymore, right? And it's a sort of a nightmarish scenario, but a lot of people like the CDC, the WHO, this antibiotic resistance problem is on everyone's mind because it is the next pandemic. And when antibiotics stop working, it's not going to be a pandemic, it's going to be every pandemic, right? Because suddenly every bacteria becomes a deadly bacteria,
right?
And so the race is on to try to identify new antibiotic molecules or antibiotics that you can use that bacteria will not be resistant against, right? So you need a next generation of antibiotics.
Where will they come from?
They can come from all sorts of places, right? The original ones came from a fungus.
That's true.
So a lot of them actually come from fungi because fungi are good at killing bacteria. But there's all sorts of compounds that are being screened naturally and things like that. We have been extracting them from this sort of cool source. It's bacteria's sort of natural enemy. There are these viruses called bacteriophages.
Yes. Yeah.
And phages are the most numerous biological thing on Earth. They're everywhere. They outnumber everything by bazillions of times, and they kill bacteria. That's all they do. They don't kill people. They don't infect people, but they kill bacteria. And they have mechanisms for doing this, right? And they're very simple. They just have a few proteins in them. So if you can pull the proteins out of the phages that the phage uses to kill a bacteria, those proteins those are basically super powerful antibiotics. And what's nice is they don't do anything else. So it's a normal antibiotic will often, one of the problems with regular antibiotics is that they kill everything, right? So you take antibiotics after a surgery, it wipes out your microbiota, it kills all these other microbes, it kills the good bugs and the bad bugs.
Yeah.
But what you really want is you want something that just kills that bug that you're trying to kill and doesn't harm everybody else.
Is that right? I heard somewhere that every bacteria has a phage against them. Is that right?
Well, it has to be that it has to be the case because, if you think about how many bacteria are out there and how many phages are out there, it's this constant universe, the universal war. So there's certainly going to be a phage against every bacteria somewhere. Doesn't mean we have them. We haven't, necessarily cultured them or sequenced them or identified them. But I think that's probably true. Is that they've done this study. So phages are so numerous.
Yeah.
So in the ocean, which is where most life on Earth is in the ocean.
Right.
There's so much sort of cyanobacteria and other bacteria, and phages kill them. So about 80% of all life on Earth basically dies and is recycled every day. And it's mostly these microbes in the ocean. And so the ocean is basically phage soup, virus soup, and it's just constantly killing bacteria. And so imagine you have you have an algae or cyanobacteria that it'll absorb carbon from the air, right? It'll absorb CO2. and then the phage will kill it, and it'll die, and it'll like sink to the bottom, and coral or whatever. And so much of that is happening that phages are actually credited with altering the global carbon cycle. And so there's so many of them. So I think it's safe to say that if there's a bacteria somewhere out there, there's a phage that.
But you have to find them, right? You have to go
find them, right?
Do the scuba diving. We have to go to caves. We have to go to deserts.
What's cool is, so with our technology, right? What our technology does is it allows you to discover phages and which bacteria they're targeting without having to grow anything. So the problem is not that we don't have enough of them. So you can go to you can scuba dive, right, and scoop some water. But the problem is you'll bring that water back to the lab. You won't be able to grow the phage,
right?
Because the phage only grows in the bacteria,
right?
And that bacteria only grows in the ocean. So you might have it, but you won't be able to grow it. And the reason why the sequencing technology helps you, it helps you discover these things because you can sequence their DNA even if they're not alive. And so you can get water from the ocean. We actually did this. We did one of the projects we published recently was they took microbiome samples from super deep ocean like hydrothermal vents, right, where there's who knows what kind of biology is going on down there because there's no light, right? Their carbon source is like whatever, or their energy source is whatever is shooting out of those vents, and so you can scoop some of that material. You would never be able to grow anything that's in there because it's evolved to grow in these crazy environments. But you can sequence the DNA, right? And when you sequence the DNA from that community, you can discover the new bacteria that are there, the viruses that are there, all this other stuff. And then you can try to understand what they're doing by computational methods, right? You can see what the
So what's the big plan here? Do you plan to have your own phage bank and then help create new classes of antibiotics? Or
so what's cool is, so every phage has a protein that's called a lysin, and that's sort of like the torpedo that the phage uses to break out of the cell. So when a phage infects a bacteria, it then replicates, and then it has to break out and to infect more bacteria. And the way it breaks out is by using this protein called a lysin. And so lysins are single proteins that kill the bacteria.
Right.
And if you just take the lysin without the phage, the protein itself, you put it on some bacteria, it kills them instantly. It bursts them.
It can target it with specificity?
Yep, and it's specific to the host because that's what the phage was targeting. So for example, you can imagine you have a bacterial culture that looks like a beer. You can put a bunch of lysin protein into it, and in 10 minutes it'll look like apple juice. There are these videos that are, this is there's all these papers on it, there's classes, but there are videos on YouTube about lysins. You can watch them under a microscope slide, like a bunch of bacteria swimming around, and as soon as lysin protein enters the space, they just go pop, pop, pop like balloons.
Wow.
Like in front of your eyes, like in seconds, you can wipe out an entire field of bacteria. So they're cool. And the other cool thing is they don't require you to grow phages. It's just a gene, and you express that gene, you make a protein, and that protein is your antibiotic. So you don't have to have phage banks, right? You can express these lysins. So I can tell you about a really cool project we're doing now. This is public information. So we're working with the Gates Foundation to because again, we have this massive database of millions of phage genomes and lysins, and we know who their targets are. So we were approached by the Gates Foundation. Say, can we use these lysins to knock down enteric methane in cows? So cows, as you know, produce lots of methane. So they eat grass, the bacteria inside their rumen chew it up, and they barf methane into the air, right? And that methane is super strong greenhouse gas.
Right.
If you there are some estimates where if you knock down this cow methane by 50%, that's the same as taking every car off the road.
Wow.
In terms of greenhouse emissions.
Yeah.
It's intense because methane is so much more powerful than CO2 as a greenhouse gas. So, but the methane is created by microbes that live in their guts, in their rumen. They're called methanogens because they make methane. And so what we're doing is we're actually taking these lysin proteins that are in our database that came from rumen samples. So we have a bunch of phage genomes that kill methanogens. And if we take those samples, if we take those proteins, we can actually kill methanogen bacteria and archaea without having to have phages. And so that's a project we're working on now, is basically to use these phage proteins as antibiotics, but not against pathogens, but against bacteria that are making methane.
That's fascinating. No, it's really fascinating what you're doing with bacteriophages. I always find it fascinating.
Bacteriophages are really interesting. There are a couple of books recently came out about the history of that space.
Yeah.
It's amazing because they've been around for over a century.
Yeah.
And they kind of show up in the public eye and then disappear. They show up in the public eye and disappear. Right now, I think they're on the up. Yeah. People are really doing a lot of work in phage now.
There is one very famous river in India. In the West, we call it Ganges. From thousands of years, people are going there to treat their infections.
That's where the phage therapy field started.
Yeah.
It was some Western explorer who went to the Ganges and was like, why does this happen?
Yeah.
And that's where some of the first phages were isolated from. And they didn't know what they were phages. They had no idea what it was. There was just like some particles. They thought they were proteins. It
was all scientific, right? From thousands of years. Even every year now, there's like a festival. They call it Kumbh Mela, where millions of people go there.
But it's because it's so infected, right? Like he went there and he's like, people who have like whatever it was, like cholera or something, or diphtheria or something, and they go in there and they cures it. So like, it's because there's so much of that toxin in the water. And so there's all these phages against these bacteria. So if you can pull them out, you can treat some of these diseases. But yes, it's true. I mean, that's what a lot in the Soviet days during World War II, possibly even World War I, there was a lot of work from the Soviet side on using phages because most soldiers would die from like frontline diseases. They wouldn't even die from bullet wounds. They were dying from from dysentery and stuff like that on the front lines. Yeah. And so they would get like corpses and isolate like what we now know as phages that target some of these pathogens, and they would like soak them on blankets and stuff like that, and it like dropped the rates of these diseases quite a lot.
Right.
In the army, that was one of the things that's credited to like helping the Soviets in the Battle of Stalingrad and stuff like that, just because they had this phage technology and the Germans didn't. Right. So it has a fascinating history how people have been using phages in the past century or so.